Hello, everybody. I'm really pleased to share with you information on how Edgewise Therapeutics is focused on muscle disease to develop a new therapeutic potentially. We are working to develop something to protect muscle in both Becker and Duchenne muscular dystrophy. Edgewise is a public company, so that we need to give you certain precautions that I am talking about things that may happen in the future. And most importantly, sevasemten, or EDG 5506, is an investigational agent that is not approved in any territory. Edgewise has been on a mission to discover new medicines that can hopefully improve the lives of people facing serious muscle diseases. We've been around since 2017, and we are focused on developing muscle-targeted therapies for muscular dystrophies. Edgewise is really the only company that is focused on the contraction-induced injury that underlies the fundamental problem in Becker and Duchenne muscular dystrophy. We're studying approaches to protect the muscle, and our goal is having an investigational agent, an oral daily pill that can be used alone or in combination with other therapies, regardless of mutation. Now, we have a number of clinical trials that are ongoing, and to give you a brief overview of them, ARCH is a 24-month open-label study. It's ongoing, and it includes adults with Becker who were in the phase 1 study, as well as others who have joined. And importantly, that has given us information from men who were in the decline phase of their disease. We've shown that sevasemten treatment was associated with stabilization of their disease for 2 years. It was also shown that there was an immediate and profound decrease in a number of the biomarkers of muscle disease. And this has given us the information to design a study that is ongoing and enrolling patients, Grand Canyon, which is in ambulatory adult men living with Becker, and that is enrolling in the United States as well as across the world. We also have a program in Duchenne muscular dystrophy. The LINK study is an initial dose-ranging study in ambulatory children with Duchenne. That is actively enrolling now. And FOX is a study in children and adolescents with Duchenne who have been previously treated with gene therapy, and that is also actively enrolling. Now I'm going to show you a video about how muscles work and to give you a little bit more insight into the contraction-induced injury that causes damage in these diseases.
How muscles work and why they sometimes don't. Our muscles do a lot of work for us. We all know they make movement possible. Every muscle is made of thousands of long strands that contract, creating movement in the body. These contractions put a strain on the system, leading to little tears in the muscle fibers. This is called contraction-induced injury, and it happens to everybody. It's why people can feel sore after exercising. Protecting muscles from too much injury is where certain proteins, including dystrophin, have a vital role to play. Ah, yes, there he is. Look at him go. These proteins connect our muscle fibers together so that strain on muscles is spread evenly when the muscle contracts, similar to threads in a rope working together to strengthen and stabilize the muscle. When muscles are working properly, tearing is minimal and repair is quick. Soon the damaged muscle is back online, doing what it needs to do. Sometimes key parts of the muscle are not working, so the muscle is susceptible to damage from muscle contraction. We see this in muscular dystrophy, a group of diseases that includes Becker and Duchenne muscular dystrophy. In these diseases, dystrophin is only partially functioning, as seen in people living with Becker. Oh, yes. Or totally absent, as seen in people living with Duchenne. That means that when muscles contract, force from the contraction isn't spread out over the muscle, which can damage the muscle fibers. In contrast, in fully functioning muscles, muscle contraction doesn't cause excessive injury. But in people with Becker and Duchenne, damage can be more frequent and severe when their muscles are contracting. The muscle membrane can become leaky and let unwanted visitors like calcium inside the muscle cell, causing a complete fiber breakdown. Over time, the muscle gets inflamed, and instead of being able to repair itself, scar tissue and fat are laid down. When this happens, we often see muscles swelling at first from the stress and then shrinking as they become less effective and are used less. This muscle wasting is also called atrophy. Damaged muscle fibers don't do their job and are weak, which puts more strain on the remaining fibers whenever they contract, resulting in a cycle of continued muscle damage. Over time, the impact of muscle damage caused by contraction in Becker and Duchenne causes loss of muscle and therefore loss of strength and function. Moving around becomes harder. Standing up and walking become a challenge or sometimes impossible, and many people need a mobility device to get around. Although there's no cure for Becker or Duchenne today, medical researchers are looking for treatments with urgency and focus.
Protecting muscle is a key goal in muscular dystrophy research, and there are many ideas being explored to protect muscle from injury caused by muscle contraction and to preserve function, to improve the lives of those with Becker and Duchenne muscular dystrophy. In the meantime, the progression of Becker and Duchenne can be best managed through working with your care team to identify options for protecting and preserving muscles. Your doctor and care team can help you monitor your disease or that of your child and build the most effective plan to manage it.
And that's how muscles work. In dystrophinopathy, the fast muscle fibers are particularly affected by that damage that you saw. And that can lead to fiber breakdown and ultimately loss of muscle, which then triggers loss of function. We've developed sevasemten, formerly known as Edgewise 5506, to protect dystrophic muscle against contraction-induced injury. Now, this is a muscle from a mouse. It's a mouse model of Duchenne. And on the left, you can see the muscle contracting at 100% by itself without EDG 5506. And what you can see develop are these blebs. You can see visibly the damage when the calcium fluxes into the muscle and the muscle is damaged by contraction. But on the right side, this muscle has been exposed to sevasemten. And even though it's contracting, it is able to do what it needs to do. It's contracting. It is not affected by that contraction-induced injury that you see in the same type of muscle from the MDX mouse. So sevasemten is able to completely protect against that visible contraction-induced damage. Now, as I told you, to investigate this further, we have a number of studies. The actively enrolling ones are Grand Canyon in adults with Becker and Lynx and Fox in children with Duchenne muscular dystrophy. Ages 3 up to 10 in Lynx, at 4 up to 10 in Lynx, and ages 6 to 17 in Fox. Now, Grand Canyon is the pivotal study in Becker. That is an 18-month study with a minimal number of visits, 7 visits over 18 months. We look at functional assessments, and this is intended to potentially register the drug. We're looking for men aged 18 to 50 with Becker who are ambulatory. There are a number of sites across the country that are enrolling. We have tried to do this to minimize the need for travel for patients and to ease the burden. And our trials in Duchenne are designed to look at the whole spectrum of boys who are treated with steroids alone, who are treated with other dystrophin-targeted therapies, Some are not treated with steroids, and those that have previously been treated with gene therapy. And in our overall goal is to preserve and potentially improve muscle function in individuals living with any mutation of Becker or Duchenne, or by protecting the most susceptible muscle fibers, and to be used alone or in combination. We are committed to reduce the trial burden by covering expenses, prepaid expenses, to minimize the burden on patients. And with that, I'd like to thank the people who have been involved in the study and give you the context to ask us more questions. Thank you so much for your time.